The MCAT, as you know, is no basic science test. You're going to see the science that you've learned from all different angles and all different forms and in many different ways. You've got to get used to that if you're going to succeed. That's what this lesson is all about. Concepts, science concepts in multiple forms. Let's get started. In this lesson, what we're going to see are two ways that the MCAT shows you science concepts in multiple forms. The first is the same science concept in different subjects or disciplines, and the second is the same kind of information in different presentation formats, like graphs or tables or text. The science topics that we'll be seeing this in the context of are oxidation reduction and gene regulation, and we'll actually be seeing them in that order as well. So first, as I said, we're going to see the same science concept in multiple disciplines. And the first science concept we're going to see in multiple disciplines is oxidation and reduction. First, in general chemistry, we will see oxidation and reduction in contexts like this. This question asks us about the formation of rust, and it gives us this reaction. We get a question about what iron does during this reaction. And if you're familiar with these concepts, you should be able to answer this question. Give it a shot. What do you think is the answer to this question? Well, as one of the mnemonics goes, oil rig. Oxidation is loss of electrons, reduction is gain. So that means in this reaction, since iron moves from elemental iron into ionic iron, notice that iron bonding to oxygen in this way means iron needs to take on a positive charge. A positive charge is a loss of electrons. That means it's getting oxidized. This is a basic way that redox or oxidation reduction might get tested in a general chemistry question. But where things can get exciting is when we see this same concept, oxidation and reduction, tested in different subjects, like organic chemistry. This question, question 2, says that aldoses, a certain type of sugar molecule, can get reduced with lithium aluminum hydride into something called an alditol. We get asked which of these four pictures, which of these four molecules is the product of the reduction reaction of glucose. Now, if we use the same logic we used in the last question, oil rig, oxidation is loss, reduction is gain of electrons, then, well, that may not get us very far with this question because it's hard to track the electrons themselves moving from place to place in any one of these molecules. That's why when you see oxidation and reduction in organic chemistry, you will more likely want to think of it as oxidation being more bonds to oxygen and reduction being less bonds to oxygen and usually more bonds to hydrogen instead. Based on that logic, which of these answers is probably correct for this question? The answer is B. We're asked for the product of a reduction reaction, and this molecule with all of these alcohol groups is the most reduced form. Choices A and C and D all have a carboxyl group with lots of bonds to oxygen, which makes them more oxidized than choice B. Notice here the connection to our learning goal. This same concept of oxidation and reduction, we have been able to answer two questions using two different conceptions of it. Question number 1 we answered by counting electrons. Question number 2 we answered by counting oxygens because in general chemistry we tend to think of reduction as more electrons, and in organic chemistry we tend to think of reduction as less oxygen. It's still the same process because the carbon in this case, when it gets reduced, has some electron density taken away from it by the oxygen. That's why it's called oxidation. But for you on test day, a different thought process can help depending on the context of the question. Let's now see one more incidence of oxidation and reduction. Here's question number 3, which shows us this concept in the context of biology. Read through this one on your own Think about what oxidation reduction usually means in a biological context like this and pick an answer. The answer to this question is D. The question itself brings us to the setting of the lungs, and we hear that in the lungs, deoxyhemoglobin turns into oxyhemoglobin. And based on the way these two words are constructed, this means that we go from no oxygen on the hemoglobin to having oxygen on the hemoglobin. Looking at the structure of words like that, by the way, deoxy and oxy, even if you're not 100% sure of what the word means, is always a good place to start. Anyway, if we're moving from no oxygen to some oxygen on the hemoglobin, well, that sure sounds an awful lot like moving from a reduced state to an oxidized state. And that's where redox really comes in. Once you've got that in mind, you can move to the answer choices. And the way this question tests oxidation and reduction in a biological environment is every one of the wrong answers actually gets this arrow the wrong way around. Choice A says the oxidation state of the iron decreases. Well, that's wrong because a decrease in oxidation state would mean a reduction, not an oxidation. Choice B says the hemoglobin gets better at absorbing red light, but that's wrong too, because if you know what happens to the color of your blood when it gets oxygen in it, it turns more red when it is oxygenated, meaning it reflects red light more, not absorbing it. Choice C says that in this process, hemoglobin has a decreased affinity for oxygen, but again, that's wrong too, because if you're familiar with hemoglobin, when it picks up some oxygen, it is then more likely to pick up even more oxygen, and in fact has an increased affinity. Choice D, however, is correct because hemoglobin itself is binding to an oxidizing agent. In this case, it's interesting, the oxidizing agent is oxygen. Now, each of these three questions we've just seen is interesting in its own right, but the important takeaway is to see them all together, because that's what the MCAT is going to ask you to do. On test day, you could very well have the concept of reduction and oxidation tested in three totally different ways. You can't just depend on oxidation always meaning loss of electrons, because if you always thought of it that way, you'd probably get questions 2 and 3 wrong. You also can't only think of oxidation as hemoglobin, because that will get you questions 1 and 2 wrong. It's about learning the multiple facets of a given subject, whether that is oxidation and reduction, things like work and energy in the Chem Phys section, or, as we're going to look at next, the concept of regulation. Now, before we see practice questions about regulatory mechanisms, let's take a little detour towards the TRIP operon to make sure we're starting off on the same page, knowledge-wise. The TRIP operon is an operon or a regulatory mechanism in the genes of bacteria. What happens is that RNA polymerase, as represented by this little circle, moves right along the gene from left to right, and if there's no repressor here on this little operator, then the gene gets transcribed. The RNA polymerase moves along, makes an mRNA transcript, and so on. The repressor itself may be there in solution, but in this model, the repressor can't bind to the operon by itself. It's represented here by the fact that you only get this little quarter circle on the repressor. It can't bind by itself. But when a co-repressor, this guy, is there in solution with the repressor, they can bind together and in fact block this RNA polymerase from moving forward. It will disallow the RNA polymerase from reaching those structural genes and transcribing the mRNA transcript. So again, this TRIP operon, which is a real operon, it's a real regulatory mechanism in bacteria, allows the gene to get transcribed when the co-repressor is not there, but it stops getting transcribed when the repressor shows up. This model is what's asked about in question number 4. Question number 4 says that the repressor is itself repressed. That means if we're taking notes on this image, this repressor is not going to be present. Then we get asked, tryptophan synthase, which the question stem helpfully tells us is coded for by the TRP structural genes. Remember, that's this area right here, the structural genes. The question asks, when will that transcription occur? Again, if there's no repressors. The answer choices follow a structure of tryptophan synthase will get coded for in the presence of tryptophan, the absence of tryptophan, both presence and absence, and neither in presence nor absence. So they follow that sort of programmatic language. Now, before you pick an answer, remember the reason why they're saying presence and absence of tryptophan in these answer choices is because we learned in the figure that this co-repressor, that is tryptophan. So will the structural gene get coded when the tryptophan is around or not, or both, or neither? What's going to be the answer to this question? Pick it yourself. The answer here is C. The structural gene is going to get read by RNA polymerase whether tryptophan is around or not. Because no matter whether or not that co-repressor, tryptophan, is there, there's no repressor. Remember, the question stem told us the repressor is repressed. So RNA polymerase is always going to be able to get over here whether tryptophan is there or if it's not there. Basically, this question has told us the regulatory mechanism is broken. No matter what, this thing gets produced. And in that spirit, let's move to question 5. Now, question 5 may look like it's totally out of nowhere, given the fact that we're talking about gene regulation. If you recognize the content in this question, it's talking about ACTH secretion and cortisol levels. That's a totally different ballpark than gene regulation usually. Cortisol levels tend to be about the endocrine system. But we'll see that the connection between question 4 and question 5 actually goes deeper than that. Let's take this question a little bit more slowly. The question stem tells us that normally having dexamethasone in the system means that ACTH secretion goes down and cortisol levels also go down. The question stem tells us that. But then there is this patient who has low ACTH and high cortisol, even though we gave him dexamethasone. And the question asks us, this patient, what's wrong with him? The answer choices say, does he have a tumor on his adrenal cortex? Does he have a tumor on his hypothalamus, his anterior pituitary, or is there actually nothing wrong with him? Well, right away we can say there's something wrong with him because they gave us the normal situation and they gave us his situation. His situation is different. But let's look at where that break happened. If you compare the normal structure with the patient structure, it all goes wrong right here. Normal and patient both got dexamethasone. Normal and patient both had low ACTH, but normally that inhibits cortisol. In the patient, he's making cortisol all the time. And out of the 4 answer choices that we have, that is most suggestive of choice A, that he has an adrenal cortical tumor. If you're familiar with the endocrine system, you'll know that the adrenal cortex is what produces cortisol, so there's probably a tumor there producing a whole lot of cortisol, whether or not there are regulatory mechanisms in place. Now hold on, what did I just say? I said that this patient is producing a whole lot of cortisol whether the regulatory mechanism is clamping down or not. That's the exact same thing that happened in question 4. In question 4, that bacterium was producing tryptophan synthase whether the regulatory mechanism was present or not. There was a negative feedback mechanism. In the case of question 4, it was this co-repressor system. In question 5, it's that dexamethasone would clamp down on ACTH and cortisol production, but that negative feedback mechanism has broken, which means that the final product always gets produced in large amounts. That is what ties these 2 questions back into our learning goal, that it's the same concept, regulation and negative feedback, in 2 totally different disciplines. Now, how can this help you on test day? Well, when you see anything that hints at a negative feedback mechanism, then you need to be able to apply the concept of a negative feedback mechanism to whatever the context is. Just like in the same way of oxidation and reduction from questions 1 through 3, anytime you see a hint of oxidation and reduction, the exchange of electrons or charges, you need to be ready to apply what you know about it. The MCAT will do this, and the main purpose of this learning goal is to get you comfortable with this and get you to value those connections when you find them. So next time you're studying and you say, oh wow, I never thought about regulation working that way before, but it really applies, I bet you that's going to come up on a practice test at some point, because those are the types of connections the MCAT tests about. Let's hit this second learning goal now. It is also about concepts in multiple forms, but unlike learning goal 1, it's not science concepts in multiple subjects, but it is ideas in multiple formats. Let's see what that means. On this page, we have questions 6, 7, and 8. And I'll tell you right now, questions 6, 7, and 8, they all have the same answer choices and they all have the same right answer. So once we find one right answer, we'll have it for all of them. But what questions 6, 7, and 8 are here to do is to show you that the MCAT can present the same information in totally different looking ways. Let's look at number 6. Number 6 happens to give us a gene regulatory mechanism. We're told that this protein, beta-galactosidase, is encoded by a structural gene of the lac operon. And there's this study where there are 3 E. coli cultures, samples A, B, and C. Sample A was given glucose at 0 minutes. Sample B was given glucose at 30 minutes. And sample C, well, we never gave them glucose at all. So here's the first question. Now that you know what this diagram represents, what seems to happen when we add glucose to the culture? When we add glucose to the culture, beta-galactosidase stops getting made. For sample A, we see that beta-galactosidase never really gets made. We're told that it always has glucose. For sample B, beta-galactosidase stops getting made at the very moment that we gave it glucose. And sample C, well, that makes beta-galactosidase all the time. So in following the Kaplan strategy for science questions, making a good prediction here, we probably want to look for a choice that says, when you get glucose, you stop making beta-galactosidase. Based on that prediction, what's going to be the correct answer here? The answer is C. It matches our prediction in that glucose decreases the expression of the lac operon when lactose is around. Those last few words are important because we are told in the question stem that the substrate contains lactose in the first place. So you throw glucose in, the lac operon doesn't get expressed anymore, which means beta-galactosidase doesn't get made because beta-galactosidase gets coded for by the lac operon. It's a cause and effect chain that is supported by the visuals we're given and the text that we've been given. But let's see now another way this same question logically could have been asked. Question number 7, it's got no picture to it, but it actually covers the same information as question 6. Let's see how. Well, first, question 7 outright tells us that cause and effect relationship that we saw in question 6. It says that in order to use lactose, E. coli needs to transcribe the LAC-Z structural gene of the lac operon, and that lets beta-galactosidase get translated. Presumably, that protein, beta-galactosidase, then allows the bacterium to break down lactose. That is indeed why beta-galactosidase has the word lactose in it. But then question 7 goes on to tell us exactly what question 6 told us in the picture. It tells us that when lactose and glucose are around, E. coli wants to use glucose. And crucially, it doesn't produce beta-galactosidase. But when there's no glucose, when it's absent, then lactose induces the expression of beta-galactosidase and the bacterium is able to digest it. This information, even though it's in text rather than in a picture, brings us to the same conclusion that glucose decreases the lac operon expression when lactose is around, because the bacterium wants to use glucose if it can, so that glucose basically shuts down the lactose digestion machinery. That, again, is exactly what we saw in the picture of when the glucose gets added, it shuts down the beta-galactosidase, which is the lactose digestion machinery. Let's see this now in one more context. We've seen this question in a graph. We've seen this question in text. Now let's see it in a table. Question 8 starts by telling us that beta-galactosidase is created by the LAC-Z structural gene. That's old news. We saw that in question number 7. But what question number 8 does that the other two questions don't do is it tells us about these scientists who made mutant strains of E. coli. What they did was they mutated a different sequence in 4 different types of bacteria. The first sequence, they mutated LAC-Z. The second one, they mutated LAC-O. Third one, they mutated LAC-I. Fourth one, well, they didn't mutate anything. That was just the normal wild type. And yes, you probably know what the right answer to this question is because we've seen it 2 times already. So we'll just give that away. The answer is C. Glucose decreases this expression when lactose is around. But this is my question for you. Where in this table is that proven? What's the detail in this table that proves that choice C is correct? The key here is all in the last line of the table. What happens in a normal bacterium, a wild type? When glucose is around, well, we don't code for beta-galactosidase. Remember, the pluses and minuses in this table are about the presence of beta-galactosidase. It says so in the question stem. So when only glucose is around, we don't make the beta-galactosidase because the bacterium want to use the glucose. But when only lactose is around, well, then they make the beta-galactosidase because they need the beta-galactosidase to digest the lactose. But here's the final key. When both of them are around, the bacterium also doesn't make beta-galactosidase. That is, in the presence of lactose, when the bacterium would otherwise have made beta-galactosidase, it doesn't make the beta-galactosidase only if glucose is also around. That's what the last row of this table tells us, and it's completely in line with choice C. Now, the reason why we've done this same question 3 times in a row, why it's useful for you on test day, is because you've now seen the same ideas in 3 different representations, in the graph, in the text, and in the table. And this is yet another way that you can diagnose your own strengths and weaknesses for test day. Think to yourself right now, which one of these 3 questions, 6, 7, and 8, was the hardest for you? Which one was the easiest one for you? Well, that is an important thing you have now learned about yourself as an MCAT test taker. If question number 8 was hardest for you, well, then you should probably pay extra attention to learning things about tables in your practice. If question number 6 was hard for you, you should be looking for figures and graphs, and you shouldn't be surprised if you have trouble with them, but that's where you need to practice and learn. That's why thinking about these science concepts being expressed in different ways is so important. So let's see this lesson in review. The first learning goal told us that we should be flexible when we hear a science concept called out. When you hear reduction or oxidation, you can't just get closed-minded and say, oh, it's always bonds to oxygen, or it's always electrons. Sometimes it can be about hemoglobin and be out of left field. You can't lock yourself into one way of thinking about a science subject when it crosses those disciplines, because the MCAT's going to demand you know all the different types of disciplines of these common science concepts. Then our second learning goal showed us that concepts and ideas can be represented in many different ways. We saw that same information, the fact that this bacterium usually produces this protein, but it gets shut down whenever glucose gets added to solution. First we saw it in a graph, then we saw it in text, and then we saw it in a data table. But there are many other ways ideas can be expressed as well. You should start being sensitive to the form that an idea takes when you see it on a practice test. And if the form that an idea takes is what made you get a question wrong, well, then you've diagnosed something about yourself, something you can improve in the future. Now that this lesson is over, you should be a little more familiar with this aspect of the MCAT, that it's going to take the same idea or the same science concept and show it to you from many different angles and in many different forms. Look out for those different forms in your study, and good luck.